everyone, now I'm going to explain briefly on the application of chloroplast genome in plant science. As far as we know, the chloroplast genetic engineering improves humans' everyday life. Firstly, it is applied in the production of vaccine antigens and biopharmaceutical. So, recent work has shown the potential of using a eukaryotic microalgae, Chlamydomonas reinhardti, which are used in the expression host instead of using mammalian cell lines, E. coli or yeast. The chloroplasts of these microalgae are chosen as it has a stable expression and already have GRAS or genetically recognized as safe status. So using microalgae as a platform for a simple, low-cost formulation is a very good choice. Other than that, there is also one successful story of genome chloroplast related to vaccine. As we all know, cholera has a high rate of mortality and have a very expensive licensed vaccine. These problems are then solved by using chloroplast genetic engineering. By fusing cholera toxin B subunit or CTB of Vibrio cholera with malaria vaccine antigen apical membrane antigen AMA1 and merozoite surface protein, it will result in the longest duration of protection. These particular genes are expressed in lettuce or tobacco chloroplast. The chloroplast genetic engineering can also be applied in phytoremediation. Phytoremediation is a bioremediation process that implements the use of plants to stabilize, remove, and destroy contaminants in soil. During the process, plant will release natural substances through its roots and supply all the nutrients that are essential for microorganisms living in the soil. Phytoremediation also have a very significant reduction of contaminated materials meant for disposal. In phytoremediation, chloroplast transformation technology or CTT are used for the synthesis of metal chelates. As for example, two bacterial genes encode for the enzymes mercury ion reductase MERA and organomercurial lyase MERB were expressed as an operand in transgenic tobacco chloroplast. The MERB gene breaks down organomercurial compounds that are the most toxic forms of mercury and cause damages in plants as it targets the chloroplast genomes. The CTT also used to improve heavy metal tolerance in phytoremediation. As an example, gene OSM-T2C from Oryza sativa were transferred into chloroplast of Arabidopsis thaliana, thus improving the tolerance to copper stress and increased reactive oxygen species or ROS scavenging ability. Next is the production of plant with anti-stress tolerance. So the stress tolerance is the ability to be relaxed and composed when faced with difficulties. It is very important for the plants to have a high stress tolerance so that the plant can survive for a longer time. Plant stress can be divided into two primary categories. Firstly is the abiotic stress and the second is the biotic stress. Abiotic stress is a physical or chemical insult that the environment may impose on a plant. The example of abiotic stress includes the cold, drought, salts, and heavy metal. So, for biotic stress, it is a biological insult to which a plant may be exposed during its lifetime. It is caused by living organisms, especially viruses, bacteria, fungi, nematodes, and insects. So, producing plants with stress tolerance is very important for plants protection and for the yield enhancements. Recently, scientists have begun to explore new strategies to improve the sensitivity of specific target gene. This is to allow the expression of double-stranded RNA within the chloroplast genome and to allow the use of RNA interference to occur so that the desired traits, mainly for the resistance to pests, can be obtained. We can take a look on the two examples, which is the pinealia ternate agglutinins and the production of transplastomic potato plant. Firstly, expressing the pinealia ternate agglutinin PTA gene in the chloroplast genome has resulted in multiple resistance against aphids, lepidopteran insects, bacterial and viral pathogens, and as well as whiteflies. Secondly, when producing transplastomic potato plant, beta-actin produced target long double-stranded RNA, and there were little to Colorado potato beetle or Leptinotarsa disseminata larvae. Okay, and then finally is the production of industrial interest. The example of industrial interest include astaxanthin, enzymes, and amino acid. Astaxanthin is a highly valuable red pigment that are processed to be feed dye, particularly as a feed supplement in aquaculture and poultry farming. As a result of chloroplast transformation technology, large quantities of astaxanthin, which is up to 5.44 mg per gram dry weight, has been produced by transplastomic tobacco plant that express both the beta-carotene ketolase and beta-carotene hydrolase gene from marine bacterium Breviodimonas species strain ST212. The second is to produce the cellulitic enzyme, which is to be precise, the xylanase. Xylanase is also one of the industrial interest product which are used in the paper making, fiber, baking, and as well as brewing. The gene ZIN-A is responsible for the expression of xylanase. This particular gene was transformed via the tobacco chloroplast genome, and then the xylanase were produced. So, chloroplast engineering can also be a brilliant strategy to enhance desired nutrients in crops. As an example, seed from soybean, rapeseed, brassica naples, and maize have a very low of alpha tocopherol content and high level of gamma tocopherol. The alpha tocopherol is an important precursor of vitamin E and have gamma tocopherol as its biosynthetic precursor. So the pathway can be catalyzed by gamma tocopherol methyltransferase in order to produce alpha tocopherol. By transforming gamma TMT or tocopherol methyltransferase gene into the chloroplast genome, multiple layers of the inner chloroplast envelope are formed. As a result, the vitamin E content will be increased.